Okay, our next big topic in genetics is biotechnology, which you might hear it referred to as synthetic biology, and also genetic engineering. So, biotechnology itself is producing products that are going to be used or consumed in our daily lives. So, this ranges from medication to our foods. You can see here in this second paragraph, it says the use of an organism to achieve a specific goal. So it can be in our foods, or it can be in our medicines, or it could even be transplanted organs. Types of biotechnology, um, simple ones, growing mold for penicillin, so that's a natural antibiotic, that's a medicine. More advanced, and this is where we get into the alteration of an organism at the genetic level. So many modern forms of biotechnology involve the manipulation of DNA, which is known as genetic engineering. So, a refresher, if we're talking about DNA, there's a few things that we have to make sure that we remember. So, the central dogma, this idea that was once a puzzle to be solved by um, several scientists that we've heard of before, uh, Francis Crick of Watson and Crick, but this group of people called the RNA tie club, what they set out to do was to determine and prove how the genetic information moves from the DNA in the nucleus out to the ribosomes to where the genetic information is translated into protein. And they figured out that it goes by way of the RNA, hence the RNA tie club. This is also more commonly referred to as gene expression. So all the information is in the DNA. It's passed along by the mRNA out to the ribosomes to be translated into protein. So all cells within an organism have the same DNA, but not all of the genes are expressed at the same time. So an example of that goes back to what you already see in the upper paragraph there about insulin. So insulin is produced in an area of the pancreas called the islet of Langerhans, but insulin is not produced in heart cells. So, one way to think about gene expression is that the gene for insulin is expressed in the pancreatic cells, but it's not expressed in the others. In fact, it's repressed. So, gene expression could be in the different types of cells, or it could be as the cell is maturing and developing. Vectors is something you're going to see throughout this. A vector is a vehicle for transmission of genes from one organism to another. Viruses and bacteria have both been used as vectors in genetic engineering. So, biotechnology, examples of biotechnology, and we're going to spend a little bit of time on all of these over the next few days. The first thing we'll start out with is gel electrophoresis, which is a process of separating DNA into fragments by sizes. And what you get as an end result of that is called a restriction map. So it's literally a map of your genes, different bands, large bands, small bands. It makes a particular pattern. No one else has the same pattern that you have unless you have an identical twin. Um, Most frequently, instead of calling a restriction map, everybody just kind of refers to it as a DNA fingerprint. Later on, we'll talk about genetically modified organisms. So this is where we get into the genetic modifications. So modifying at the level of the DNA, using those vectors to move genes from one organism to the other, and creating these genetically modified organisms that are referred to as transgenic. So they have genes from other species. And then gene therapy, how to be able to remove defective genes and to replace them with healthier versions. And we'll talk about something else called CRISPR, CRISPR. So gel electrophoresis is a process. I think they do this upstairs in AP and probably VHCC biology. Um, it is a laboratory technique used to distinguish DNA fragments of different lengths. It literally is a contraption that is full of a gel. It's called an agarose gel. And it does use electricity to separate the DNA fragments. So electrophoresis itself, we break everything down by prefix suffixes. Electro referring to electricity and phoresis referring to carry. Now this can also be done with RNA and with protein. So this is a much watched video, so I would really like for you to watch this video from the Amoeba Sisters and then come back to me.